Hello guys, let's begin with a new topic today. Today we'll talk about origin of replication and how exactly the initiation happens both in the eukaryote as well as in the prokaryote. So this is a very interesting topic. One thing I'll make sure is that whenever a cell is ready to divide, during that time you need DNA to be doubled up, whether it's a prokaryote or the eukaryote. In the eukaryotic system has evolved in such a way it has well-equipped cell cycle where we have the different phases. The S phase is one where the DNA is going to be replicated. This replication and this phase of the cell cycle both are well connected, and one triggers other. We'll talk about that in a moment. And similarly, when a bacteria or the prokaryotic cell is tend to divide, it is also going to double its DNA content, though they do not have a specialized cell cycle. I will not say it will not have a cell cycle. It has, but that cell cycle is not as defined or as pronounced as it happens in the case of eukaryote. So prokaryote do have the cell cycle, but that is not very clear. So let's first begin with the how the initiation process occur in the prokaryote, and then we'll go to the eukaryote. Let's go into the those cycle and we'll try to figure it out how exactly it is going to happen. So before I go into that, let me just show you the few things, and then we'll just try to make sure that how it is going to happen. So this is how the prokaryotic cell is going to happen. There's a certain molecules. Let me introduce you once, and then I'll define it one by one. So few protein and the kind of genes that are going to be made from. One is a helicase, that is the product of the DNA B. The loading helicase in primase, it is the product of DNA C. Single stranded maintenance protein or the single strand binding proteins we call SSB. The priming, a primer making protein is the DNA G. Sliding clamp, which is a part of DNA pol 3, which is having the 10 subunits. One of them is the beta, and the clamp loader, which is basically having ATPase activity, is a gamma and delta complex. And the basic enzyme out of the three, the one which is going to have this process to carry on further is the DNA pol 3. DNA pol 3 is always presided over by other two on the other two, pol 1 and pol 2, because it has a great processivity. It will not leave the DNA template for a long time. And then we have two enzymes which are working simultaneously. There's a protein which work as a dimerizing unit, which is going to join the two DNA pol 3 together. Then the pol 1, which is once the process is done, the removal of the RNA, the primer, because we know DNA polymerase cannot start DNA synthesis from scratch. It needs to have something because it is the one which is going to form the phosphodiester bond between the two nucleotides. And it will use the ATP, GTP or the TTP or the CTP, where the pyrophosphate will be released in the process, and that energy is going to be utilized by the DNA polymerase. So as such, it is not requiring any kind of energy from the outside. That is not the today's topic. And the one which the on the Okazaki fragment, the lagging strand, which is going to join the various fragment of the DNA on the lagging strand, will be joined together with the help of DNA polymerase. Let me just have other some the genes like pol 1 will be coded by the pol A gene, pol 2 will be coded by the pol B, whereas the pol 3 will have the several genes because it is a huge protein made up of 10 subunits. Okay, it's a the polymer of a polymer. Different proteins will be substituting that. And then we have the initiator protein, which we call the licensing factor. It will bind to the origin of replication to initiate the proceedings. Then we have the IHF, which is another DNA binding protein, which is going to bind with the origin of replication site because of oriC in the E. coli. Then we have the FIS. the fish, this also binds. They have a different purpose to be solved. Will not discuss in today's lecture. Then we have the helicases and primases, that is the DNA-B. Similarly, I told you about the DNA-C and DNA-Z. And at the same time, the last one that I didn't tell you is that, the gyrase A and gyrase B, which is a kind of topoisomerase 2, which is going to cut the DNA on the both the strands to release the strain. So let's begin, this process. Let me first show you exactly how this is going to happen. If you look at the origin of replication, there's only one origin of replication in the case of prokaryote, which is, the distinguished, by the two important region, 4 9 Mers and 3 13 Mers. So if you see in the beginning, this is, the 3 3 13 Mers and 4 9 Mers. That is the binding site where exactly the DNA-A, which is the initiator protein, will come and bind to. So what it will do is that, first it comes and binds to the DNA-A, will bind to the 13 Mers, that the 3 13 Mers, in a cooperative manner. Means the first binding of the DNA will be easy, most difficult one. It will facilitate the further binding of the DNA protein. Once all the 13 Mer are bound to the DNA-A, then it starts tilting and it starts separating the DNA on the 9 Mers. And this, allows DNA-B and DNA-C to come over there and, load the DNA helicase in the ATP bound manner, because DNA is a stable structure and nothing can be loaded on the DNA without using the ATP. This is one thing. Second thing is that, it should have the specificity. It is a pure DNA protein, interaction. Otherwise, this process is not going to take place. Now, if I show you this actually I'm less likely to talk about it, but I just want to show you what exactly is the core enzyme. If you see this is the 10 unit of the DNA polymerase 3, which is the main enzyme, and you can see the clamp loader over here. All these units, you can see the beta, you can see the delta, dash and all. And this is the 3, which is, shown in the pink: alpha, epsilon and the theta. These 3 make the core enzyme. Core enzyme is one which is going to catalyze the reaction, and other they're going to play, important role in the the loading of the other proteins together. Fine. So if you see the DNA binding sites, which are loaded by the these initiator proteins in the ATP dependent manner, and then the initial melting will be done by initiator proteins. Then helicase will come into the picture, will start opening of the DNA in the ATP bound manner or ATP dependent manner, and then it is going to start making the replication fork. This is a very simple the origin of replication and the concept of how exactly the initiation of the DNA replication occurs in the prokaryote. But there's a few more points which is very, very important. I'll show you is that in the case of prokaryote or the bacteria, the DNA replication is not complete when the next division occurs. So in that case, one thing is ensure that both the end hemimethylated region will not be allowing the other proteins to bind to it. So they have a lot of GATC regions, and that region is bound with a protein because of sec-A. Sec-A is going to occupy these regions unless the replication process is complete, and then only the methylation occur on the other side. This is how the DNA polymerase will come to know which the strand of the DNA is the new one, and what is the DNA which is the older one. So this is a very, very important process which is being facilitated by the sec-A, and this methylation process is going to be done by the diamethylase or dimethyl transferase enzyme. Now let's talk about the eukaryotic system. If you look at the eukaryotic system, unlike the prokaryotic system, here the DNA is going to be linear. And another important point is that they have so many origin of replication. So they remain bound by the ORC or the origin of recognition proteins which are present over there, and those complexes will bind to the those particular regions, especially the size of those origin of replication is smaller. We have all these information from the little organism like the Saccharomyces cerevisiae or some of the cell line or SB40 virus. Here the first protein to come and bind to, that is the CDC6, cell division cycle 6, and CDT1, they will bind, or on the origin of replication site, and then it is going to allow the binding site for the MCM, which is a kind of a mini chromosome maintenance, which is a hexameric protein which work like a helicase. And together, if you look at this, this is going to work as a pre-RC, pre-replicative complex. And this process will not go further. If you look at this much happen in the G1 process. This is one of the steps in the cell cycle, followed by the S, G2 and M. Now the S process is going to be very important, where exactly the DNA replication takes place. In this case, what will happen is that the cyclin dependent, CDK will come and phosphorylate the CDC6, which leads to the degradation of the CDC6 and the removal of the another protein, that is the CDT1, which is going to be further inhibited by the geminine protein. Now this ORC complex is going to be phosphorylated, and the helicase, that is MCM, will start the proceedings that is going to open up the DNA, and the DNA polymerases will come over and will take the charge. If you see this another diagram, you look at this, what happened on the early G1, the ORC remain occupied, followed by the CDC6. Here the CDT1 was not shown, and that will facilitate the binding of the MCM, which is the helicase. And S phase, once it is over, DNA got replicated, and again this ORC is going to be again occupied. This is well shown over here. And see this, very important, when the CDK activity is low, so no PRC formation is going to happen, or the no pre-RC is going to be activated, and formation is allowed. So formation is allowed and activation is not allowed. On the other hand, when the activity of the CDK is high, here the pre-RC is going to be activated, but new pre-RC will not be allowed to be formed. This is how the regulation is going to happen in the case of eukaryotic system. This is how I told you the cell cycle and the DNA replication both are connected to each other, and this is how the entire process. If you look at this, then the G1 phase, no pre-RC activation occurs because CDK activity is very low. Especially, we know all the cell cycle phases, they have the specific concentration of the cyclin. The phase which is going on, that cyclin is going to dominate, and the phase which is the coming next, they have at the basal level. Look at over here, when the CDK activity, especially the cyclin dependent s-cyclin and the CDK complex is going to come together, this is going to be activated, and during G2 and M, just ORC remain bound. There will be no pre-RC formation. And just give you the brief, the eukaryotic replication is not well understood, but whatever the information that we have the few enzymes that are going to play a very important role. For example, the alpha, which is going to initiate the process, delta and epsilon is going to be involved. Delta is going to work for the lagging strand, and epsilon is going to work for the leading strand, whereas the gamma, beta and other, they will involve in the DNA repair process and the mitochondrial replication. And here the helicase, as we see in the case of E. coli, it was the DNAB, as I told you in the beginning. Here the MCM complexes. So SSB is going to be replaced by the RPA. Priming is going to be done by the DNA polymerase alpha, and the clamp role is going to be played by the tcNA, and the clamp loader is going to be RFC. And the catalysis, as I told you, delta and epsilon, one for the lagging strand, another for the leading strand. And the RNA removal, which was a basic work of the pole 1 in the case of prokaryotic system. Here it is going to be the FEN, flap endonuclease 1. And the ligation for the Okazaki fragment is going to be done by the ligase 1. Hopefully, you'll be able to understand the today's lecture. and hopefully you enjoyed. For any query, you can put your comments in the comment box. I'll try to answer all those questions. Thanks for listening patiently. Thanks a lot.